Thank you, Yasuo, for inviting me to talk in this session. And I am actually really happy to see so many people on the last day who are still interested in all of these science talks. And this is my second talk at the meeting, so that's fantastic. Today, I'm going to tell you about the host engineering group that the host engineering work that my group does as part of JBay. So I wanted to give you a little bit of a background. JBay is one of 4 bioenergy research centers funded by the Department of Energy. And our mission is to establish the scientific knowledge and the new technologies that are required to transform the maximum amount of carbon available in bioenergy crops to biofuels and bioproducts. Am I speaking too loud? Okay. Okay, so that's our sort of overarching mission. And we have 11 partners, all located in one location. They're spread across the country, but more than 90% of us are all co-located on one floor of Emory Station East, which is a branch of Lawrence Berkeley National Lab, just across the bay in Emeryville, if you should ever come and want to visit us. And we've been there since 2007. So the work that I am presenting is a small wedge of what these 200-odd people have done across more than 10 years. So it's really a very, very small portion. Okay, so in order to achieve this goal of transforming bioenergy crops or other sustainable material through deconstruction using a microbial platform to a fuel, we have 5 divisions. We have the feedstocks division that focuses on plant engineering and understanding plant cell walls. We have the deconstruction division, whose work is to develop strategies and approaches to break down this plant material, including approaches that Kevin just described. Michelle O'Malley is a team member at JBay. And then finally, taking these intermediates that you get from plants and convert them to biofuels and bioproducts. And underpinning all of this work that we do are the technology and the sustainability division, which provide all the tools and analytics, as well as provide us the life cycle analysis, the techno-economic analysis, and the agronomic relevance of what we are doing to make sure that this whole work that we are doing falls into some relevance of what is really needed to solve this really big challenge. The work that I'm going to describe to you today falls squarely in this whole intermediate to final product section, the biofuels to bioproduct section. And so I wanted to start off with giving you sort of a glimpse of all these molecules that we've targeted over the years and we would like to target that are biofuels and bioproducts. Pamela described this really nicely. What is a biofuel? They're usually these featureless, non-functionalized hydrocarbon-type compounds, shorter with some branching, maybe an oxygen or 2 for the gasolines, longer chain for the biodiesels, and the kerosene-like things for aviation fuels. Bioproducts tend to be a lot more functionalized. They have heteroatom, aromatic groups, unsaturations, and so on. But the point I'm trying to make with this slide is, these compounds, many as they are, there are thousands more out there that are desirable and that we can actually make. Any one of these compounds, give it to a good metabolic engineer, they'll be able to develop the pathway and you will have the pathway. So which one should we focus on? We have 5 years, a good bit of funding, but not unlimited. How do we come up with the criteria to select these compounds? So that's actually the first challenge that we have. How do we figure out what are the most useful compounds to make from a scientific point of view that can be obtained from all components of the biomass? And the second thing is, even though we can do this work very, very well, strain engineering, development, and discovery of pathways still remains pretty anecdotal and time-consuming. Getting commercially meaningful tighter rate yields are rarely demonstrated. And this is a very important gap between the research we do and what will finally become useful to people out there. And then it's really hard to preemptively predict how a particular strain will perform when it is scaled up. Okay, so in order to meet these challenges, our goals are we're not going to target one compound just for the sake of targeting that compound, but rather we want to develop this broad platform where you can kind of plug in what's your carbon source, what do you want to make, and figure out what the best routes to make it, and then kind of develop those new technologies and tools that I said our mission was all about. We want to develop microbial hosts that will maximize the utilization of the carbon. And we want to develop predictive tools to systematically increase tidal rate yields and also improve our understanding of the scale-up. So to give you a little bit of a perspective, all of us are microbiologists, all of us have been in the wet lab at some point. In your mind's eye, visualize a microbial cell culture. It's this beautiful, translucent LB broth, and it clouds up over a few days, and it's perfect, and it smells yeasty, and your bug is growing. So that's what So keep that thought in your mind. So now with this thought in mind, I wanted to tell you that in fact, having your organism perform in this optimal manner is tied to everything that you're trying to do in this project. This microbe needs to consume every biomass component. It needs to tolerate any inhibitor that comes from the biomass component. It needs to have a pathway either natively or you have to engineer it to go from that component to your final product or some precursor. It has to have good flux through through a pathway to do so, not just a little bit. It has to maintain the right metabolism to do this stably. It has to tolerate the final products that it's making. And maybe if you get all of this right, we have a better chance of putting it in a bioreactor where it actually has to perform. So now I want you to go back to this image that I put in your mind of this beautiful cell broth. And I wanted to show you this. This is from the lab of my collaborator, John Gladden. And here he has taken some milled biomass. I'm not sure what it is. Could be eucalyptus, maybe switchgrass, sorghum. Looks like mud. And he dissolves it in reagents that can completely dissolve these biomass. In this case, it's an ionic liquid called selenium lysine. Before mixing with these reagents, it looks like mud. After mixing with this reagent, it looks like wet mud. And then when you add the enzymes to it to saccharify it to release all the sugars, it looks like gooey mud. It's basically the consistency of peanut butter. And so now here is the real, where the rubber meets the road. Your organism has to perform in this muddy, gooey, heterogeneous environment where it is being churned with great mechanical stress and you're trying to put oxygen through it. When someone says bubble oxygen through your culture, in your mind's eye, how do you bubble oxygen through peanut butter? So here's what we're dealing with, right? So it's a very interesting environment, and I think there's a lot of basic science to be done to understand how microbes perform in this environment. There's another reason I wanted to mention this is because we are actually able to study this. It is not beyond the realm of basic science because of other DOE user facilities like the Advanced Biofuels and Bioproducts Development Unit, the ABPDU, that gives us access to large-scale reactors, biomass that has been pre-treated in the way we want it at scales that are relevant, so you can start doing basic science in these sort of industrially relevant conditions. So going back, it's a good segue. I showed you that big gooey pot of material that had been pre-treated with an ionic liquid. Anything that can kind of take apart your biomass in that kind of effective manner, you can imagine it has some downstream impacts on the system you're going to expose it to. It contains toxins related or inhibitors related from the biomass itself, and the reagent itself might be toxic to the organism. So how does that translate, or what impact does that have in this final place that you want to put your organism? So here are some ionic liquids we use. Imim chloride is an imidazolium-based ionic liquid. It's the gold standard in the industry. And selenium lysine are more less toxic, more sustainable ionic liquids that JB is exploring. They both have issues with downstream enzymes and microbes depending upon which enzymes and which microbes you want to use. So how do you address this problem? So over the years, because this was a very important reagent for JBay, several groups at JBay have worked hard to develop sort of routes to overcome this ionic liquid toxicity, and you can approach this from many different angles. And here I'm listing some of the papers where you've developed like an ionic liquid tolerant host itself. Why is this an important approach? So I wanted to show you one example. So a while ago, a scientist in the group sort of overlaid some E. coli strains with ionic liquids and just let it sit there. After a while, she saw some spontaneous mutants come up on that plate. So we took advantage of our collaboration with JGI, and we just resequenced this genome. It's just E. coli, one spontaneous mutant. We didn't even know what we would get out of it, but what we got back is basically a single meaningful SNP. When you look at this, it was for a regulatory protein called RCDA. RCDA is known to regulate quite a large number of targets, and amongst them was this membrane protein, which we ultimately showed was sort of like the cause of an RCDA deletion to be able to give this sort of a spontaneous mutant function. RCDA was repressing this transporter, and this transporter was very good at exporting the ionic liquids. So what the RCDA mutation gives you is, if you will, a host chassis now that is tolerant to ionic liquids. How do you use that? Why is this important? Well, what happens is that now you have a host chassis that is intrinsically able to export out this ionic liquid that is toxic to it. So now when you couple that with your pathway can operate in an ionic liquid-free environment, even if the culture conditions contains these reagents. So how does that translate to our work? Well, we went down to the ABPDU and asked them if they would make us a big amount of ionic liquid pre-treated biomass, and we cultivated our limonene-producing organism. Limonene is a jet fuel precursor in this hydrolysate. And very simply what we saw is that actually both the wild type and the RCDA mutant, when you have this limonene pathway in it, can produce equivalent levels of limonene by themselves. But when there's ionic liquid present, the wild type can't even grow, so there's no production of limonene. But this RCDA chassis, it can grow and it can produce limonene. So now you have a chassis where you can produce limonene. You don't have to remove the ionic liquid per se. How else can we use it? Well, one of the things that the ABPDU had done is that they had added the enzymes and saccharified this biomass for us. Meanwhile, our collaborators at JBay had worked very hard for years to develop ionic liquid tolerant cellulases. So we said, we have these ionic liquid tolerant cellulases, and now we have this ionic liquid tolerant chassis. Wouldn't it be nice if we could combine one of these cellulases that works at lower temperatures that E. coli works in, and combine all of it? So we did. So now you have a chassis that is ionic liquid tolerant. It can harbor your ionic liquid tolerant cellulase and your pathway. So what you can do is you can just take your switchgrass or sorghum or whatever, you can deconstruct it using an ionic liquid reagent, and you can add it to this organism, which is now able to take at least the cellulase component of it, break it down into sugars, and convert that to limonene. So why is this important? This is important because this is what sort of like a cartoon workflow looks like. You have your biomass, it has to be transported to a plant where it is pre-treated, in our case using ionic liquids, maybe in somebody else's case using some other deconstruction method. Then because these deconstruction methods are toxic, you have to clean it up, you have to put it in a second pot where the enzyme action is done for sacharifying, and then you have to clean that up and now put that in a third pot where the fermentation, so to speak, happens. And each one of these units is money and operation and capex and opex and all of that. And so if you have a chassis that doesn't require cleaned up material, you can eliminate maybe one, maybe even more, and consolidate this entire thing into one pot, which is actually very meaningful for the techno-economics. So we did this work and we published it a couple of years ago, and it got a lot of attention, a lot of attention. I think it was just because it has such a diverse authorship across like 5 different countries. There were people, 3 countries in Europe, Asia, and so on. And so I think the whole world loved this project, is my guess. It even got picked up by a popular science book called Soonish, where they have described 10 emerging technologies that'll improve and or ruin everything. So I'm really, really hoping it's the former and not the latter that will happen with our technologies. So, okay, so that was one example. Here's another example of how host engineering is important. So here I wanted to show you an example of developing a pathway from the host point of view, So I'm using a pathway that is very, very well established. This is work by my collaborator at JBay, Taek-Soon Lee. He has worked for years on this pathway. He knows it in and out. He has figured out every single bottleneck and just refined it to no extent to make isopentenol a gasoline in E. coli. So the question is, what if people don't want to make it in E. coli? What if you don't want to feed it glucose? What if you want to use a host that uses a different carbon source? How easy is it to take this very well understood pathway and go from E. coli to a different organism? So we did this in Corynebacterium. Why Corynebacterium? Well, it's already an industrial host. People have already scaled it up. It's used to make a lot of different commodities, and one of our collaborators had shown that it is actually pretty good at catabolizing aromatic components of the biomass, and in our hands we found that it was tolerant to isopentenol, which tends to be toxic, and it is also tolerant to a suite of ionic liquids that people at JBay was interested in looking at. So we said, okay, let's try this. So this is the pathway. Anybody who's worked in the isoprenoid pathway, this is like very, very well established. These are famous enzymes. And so routinely in E. coli, you put it in a plasmid, you put it in there, you induce it, boom, you get the production of your isoprenoid. Everything is great. You put it in Corynebacterium, nothing happens. It's nothing. It's just nothing. Okay, so there was that. So I wanted to point out that the person who did all the work is sitting right there in checked shirt, Yusuke, and he had a very nice poster on this yesterday, unfortunately, or I would have pointed you to that. So what was the next thing that Yusuke did? He said, okay, let's check if the pathway is being expressed. So this is something that I will tell you, if you're trying to express a pathway and you're having problems, do some proteomics. Figure out if the pathway is expressing or not. That is the first place that problem happens. So what we saw is this. In E. coli, we were using LB. But LB is not a good media, apparently, for glutamicum. It grows fine, but it doesn't express anything. Nothing. None of these pathway proteins are being expressed. M9 either. However, if you use this defined media that has been developed for Corynebacteria that has a very different carbon-nitrogen ratio, suddenly you start seeing blips of the protein. And now when you modulate the amount of carbon source you give it, in this case glucose, you see the expression of the pathway. And once you can express the pathway, you see the production of the compound. So that's great. We figured that one out. We also found that one of these enzymes, HMGR, the level of isopentenol tracked pretty directly with it. More HMGR, more isopentenol. So what we did is we tried to optimize that, one, by putting it on the genome and stabilizing the expression. So defined media, need the right carbon-nitrogen ratio, need HMGR expression. So we also chose a different homolog of HMGR that has different reducing equivalent needs, and suddenly we were producing grams per liter of isopentenol in Corynebacteria, starting from, as I showed you, nothing. So this is like an infinite-fold improvement in production. So we had no trouble publishing this at all. Actually, I lied. There was a lot of trouble. But yes, 2 journals, 10 reviewers later, we published this. And we demonstrated that because we knew exactly how to cultivate Corynebacteria, what the growth requirements were, we could now produce isopentenol in Corynebacteria from hydrolysates. Okay, so the next example, you have to have the right metabolism. What does that mean? So for this, we turn to a yeast system. This is a very famous system. This is budding yeast, and it has something called a Crabtree effect. You all know this. So even in the presence of oxygen, if there's too much glucose present, it has this overflow metabolism where it will start fermenting and producing ethanol. And only when it respires does it go through the TCA cycle. So we wanted to show what is the impact of this sort of bifurcation in metabolism on compounds that you want to make in this organism. And so for that, we wanted to couple the choice to go down the blue line into the TCA cycle with a final product, and that's kind of useful. And to show this particular dependence, we picked something that comes off of alpha-ketoglutarate, glutamate, glutamine, all the way, just a simple 2-gene heterologous pathway producing this compound called indigoidine. Why did we choose this? Well, indigoidine is a very nice representation for a very important class of compounds called non-ribosomal peptides. Just like PKSs or polyketide synthases, they can give you a large diversity of compounds and may give us access to new biofuels and bioproducts that are not possible through other pathways. Also, this is a very nice review that I came across, not ours. Indigoidine is blue in color. It's a brilliant blue in color. So now we are coupling flux through the TCA cycle to a blue output, which is kind of nice. I don't have to tell anybody who's done mid-chain hydrocarbon analysis on GCMS what it means to have a blue output. Okay. So here I'm showing you our results going from glycerol, where it's only respiration, all the way to high levels of glucose, where it's mostly fermentation. What happens when you cannot ferment and you're respiring? You make large amounts of indigoidine right from the beginning, and then as you go to low levels of glucose, you're still making indigoidine, but then as you go to high amounts of glucose, you start getting a lot of production of ethanol and acetate, and you make no indigoidine. So this is a very good demonstration. It's the same organism, same genetics, same everything, and you get your media wrong, you're not going to get any production. So we use this to optimize how much glycerol versus glucose you could use. You know, glucose is still the better carbon source because if you have the right amounts, you get higher amounts of indigoidine. But if you add too much glucose, you go into fermentation. So using this, we mounted a bioreactor demonstration with the ABPDU, and we could demonstrate that high amounts of glucose for the same strain would just give you a bunch of ethanol and not much else. But the correct amount of glucose, when maintained with the right levels of oxygen transfer, give you a nice gram per liter of indigoidine in a bioreactor. So that was very nice. So the last example I want to show you is what if your host could do a lot of this stuff by itself? Should we select that host? What does it mean? So what if you have a host that actually consumes a lot of the biomass and can maintain the right metabolism, doesn't have this overflow business going on, can tolerate a lot of these toxins in the biomass, and doesn't have any issues with the final product toxicity? What do you need to optimize for a host like that, and what impact does that have on scaling up? Okay, so for this we chose an oleaginous yeast, a basidiomycetes called Rhodosporidium toruloides. It has very high flux through acetyl-CoA and makes a lot of carotenoids as well as lipids. So we wanted to see how that would work in our hands because it had all these other features going for it. It could utilize a range of these aromatic compounds that Kevin told you about also that come from plant biomass. It could grow in mixed carbon sources, and it did not have any Crabtree effect. So what happens when you couple that to indigoidine? So what we did is we coupled, what we did is we took our very simple sort of 2-gene pathway and we used agrobacterium-mediated transformation to transform that into Rhodosporidium, and we got a bunch of blue colonies. We picked a couple of these colonies, and unlike Saccharomyces, there was no, overflow dependence because there is no overflow mechanism. And now you didn't just have pale blue colonies, you had like deep, deep blue colonies, and they were so blue that they looked black. But there were other dependencies. You really did. This organism really likes its carbon-nitrogen ratio. You cannot go too much to one side or the other. And very interestingly, not all nitrogen are same for it. It prefers certain nitrogen sources to the other, and we're still figuring that one out. And that might have specific bearing to this compound that is actually a nitrogen-containing compound. So this is sort of like the final sort of data slide I will show you. We engineered Rosporidium toruloides to have this indigoidine production pathway. Knowing all the parameters that are required for it, we could mount a bioreactor campaign, and we started making upwards of 85 grams per liter, reaching 100 grams per liter, which on any given day is 100 to tens of thousands times better what people show for most other metabolic engineering projects. So I've shown you that these components kind of be the difference between being able to see performance at scale. And I hope I've shown you not just these anecdotes, but you can organize these learning principles into generalized approaches of how to select your host, how to develop your pathways, and how to optimize around process conditions, and what kind of data to collect to really go into this space where it can see the light of day in some real life experiences. And I wanted to, at this time, also tell you that a lot of this data we collected, we maintained through the JBay experiment data depot that makes it that much easier for us to mine the data. We have a lot of workflows going through JGI to do both synthesis and sequencing of our engineered strain, and a lot of this work was done in collaboration with ABPDU to do the work. Okay, and this is the team. Most of the work that I showed you today was done by Mara and Fayez, who did all the yeast work, and Thomas and Yusuke, who did the Corynebacteria work. There are a couple of alums to mention over here. Murray and Florence did the one-part consolidation work. I thank PER for funding, and I'll take any questions.
Great presentation. We have a question, time for one question.
One question, make it good.
Got the privilege.
So that was very nice. Regarding this blue compound that you're producing and in the final organism, you were mentioning 85 grams per liter. How is that related to the theoretical maximum yield?
Yeah, so calculating theoretical maximum yield is a very blunt thing, right? Anyone who's done it, you just put glucose on one side, compound on the other side, arrow, do the stoichiometry. But that's not what's happening here. It has nitrogen in it and so on. So it's a little bit more complicated to understand the theoretical yield. What I will say, for about every gram of glucose that is being consumed, about 0.9 grams of indigoidine is being made. So the yield is pretty high, and the organism doesn't seem to care.